Welcome everybody. Welcome back from March break. Hope everybody had a good March break. We're going to get right back into it. We're almost done our first unit. I'm thinking we're going to have our unit test a week from today. Okay, so I'm going to be giving you some stuff back. I'm going to be doing a lot of marketing this week. I'm going to give you an interim report this week on Wednesday. So I'll send you out, what your present mark is. But I kind of want to get your labs as well as your quizzes marked so I can give you kind of an accurate reflection of what's going on in Unit 1. But today we're going to go back and start talking a little bit more about enzymes. We're going to do a little activity today. So I'd like to take a look at, first I'm just going to pull up a PowerPoint here. And we're going to go through this. Okay, it's not very long, and I'll give you, a full link to this, but I'd like to talk about a few things here. Okay, so the main thing we're going to be talking about here is reactions and the enzymes that control them. Because whenever we have reactions inside our body, the main reason that we're going to, or the main way we're going to be able to control them is with the enzyme. So here's what we call like a reactant, maltose, okay, and then we're going to turn it into a products of two glucoses. So we're going to change maltose into two glucoses, okay? And then so that's the reaction. Now that reaction is facilitated by what we call an enzyme. Okay? Now the enzyme doesn't take place in the reaction. Okay, this is a glucose and this is a glucose. The enzyme is not now part of a product or anything like that. All it does is facilitate the reaction. Right? So the importance of enzymes. We've talked previously about what is required to build a cell. That's what we started with, the macromolecules. We then discussed what those macromolecules build inside a cell. Why do we need them? Well, we need to build mitochondria and plasma membranes and Golgi apparatuses. Those are our organelles. And we build those out of the macromolecules, the lipids, the proteins, the fats, and the nucleic acids. We then discussed how things are going to get in and out of the cell, and that was kind of like the cell transport, like how is oxygen going to get in, how is CO2 going to get out, how is glucose going to get in, and that was our next unit. Now our next step is going to be, well, now we can get things inside the cell, what are we going to do with them? We're going to perform some reactions. So we have briefly mentioned that we already have some biochemical reactions, but we kind of just touched upon them. We talked about neutralization reactions, condensation reactions, hydrolysis. Now we're just going to talk about, exactly how those happen. Okay, so we're going to explore this a little bit further. So this is kind of the basics of a reaction, a biological reaction anyways. So here we have a substrate. Now the substrate is another word for like a reactant. Okay, so this could be like a glucose molecule. It could be an ethanol molecule if you've been drinking booze. It could be a poison that, say, a mosquito came and injected you with some kind of poison. It could be anything. Okay, so that's going to be our reactant. And over here, we're going to turn it into something new. Okay, we want to take ethanol, which is like a poison. So after you drink a beer when you become 19, okay, you can break down that ethanol because ethanol is a poison inside our body. We got to convert it into some other things. So ethanol would be the substrate, and then we convert it to, well, there's a number of things, I'll talk about those a little bit later, that we convert them through, and we're going to use enzymes in order to do that, in order to make that reaction happen. Okay, and again, the enzymes don't take place in the reaction, they only facilitate it. Okay, so what exactly a reaction is? I'm going to talk a little bit about reaction basics. A reaction is when an electron is transferred from one atom or a molecule to another atom or molecule. Okay? This usually results in the formation of a new type of molecule, not always in some redox reactions, but almost always you're going to get something brand new that you didn't have before. So here's an example. I've got a sucrose, so this would be my reactant or my substrate. And I'm going to go through a reaction and I'm going to get something brand new. I didn't have glucose and fructose before as individuals. I had them linked together, and when they were linked together, they're considered sucrose. So these are brand new, okay? And you should almost always have something brand new when a chemical reaction happens. Right? So there's our arrow indicating that this is being converted into here. And if we wanted to, we'd usually write a catalyst over top here. I'm going to show you that in the next slide. So in a reaction, we have reactants. And again, reactants are on the left-hand side of the arrow, always. Okay? So we've got two reactants here. We've got a sucrose and water. So we've got two reactants. Okay? They're going to be converted into two products, glucose and fructose. Okay? Now, the catalyst is usually written over top of the line. Okay? It's not written where the products are because that would indicate that it's part of the products, and it's not. So we write them right over top of the arrow. So here is written as enzyme, and enzyme is used as a catalyst. So here's kind of another picture of it. Here's my sucrose. Here's my reactant. Okay, I should kind of have water in here too, but I don't. So we've got, here's my reactant. Okay, it's going to be converted into my products. And right here would be the catalyst or the enzyme doing it. In this case, it would be sucrase. So sucrose is the reactant. Sucrase is the enzyme that's going to break apart it into glucose and fructose. So this is the big thing, so the most important. When living things want to control a reaction, there's a problem, as the reactants are always available, but we may not want this reaction to happen. Say we have some glucoses in our body, but we want to keep them as glucoses. We don't want to convert them to maltose. Okay? The problem is that glucose is always going to be there. So how do we prevent it from starting to convert to maltose if we don't want it to be converted to maltose? And then other times we want to be converted to maltose. What if we have high blood glucose levels and we want to start turning it into into glycogen? Okay? We want to be able to control when things happen and when they don't. And usually, like in the outside chemistry world, we would just eliminate these. We'd say, okay, we'll take the reactants away if you want the reaction to stop, and if you want the reaction to go, put some reactants in. Well, we can't do that in our body because the reactants are always going to be there. So the only way that we control them is basically by this enzyme here, okay? That would control basically turning on and off reactions. So I want you to highlight this. You may want to write this down. Okay, this is a very important statement here. Enzymes can turn on, they can turn off, they can upregulate, and they can downregulate reactions in living systems. And almost all reactions in our body, almost every single reaction, will use an enzyme to control when and to what degree the reaction is going to happen. And what I mean by to what degree is I can turn the reaction on full blast, I want it going 100%, or I can turn it on 90% or 80% or 70%. And that's what I mean by upregulating or downregulating. Because sometimes we want to completely turn them on and completely turn them off. Other times we want to just kind of turn them down a little bit or turn them up a little bit. Okay, so we're almost done here. So we have to perform reactions in our body to stay alive. These reactions range from metabolic reactions, which we're going to talk about in our next unit, and it's going to be our whole unit is basically metabolic reactions. But we also have other reactions, like the elimination of poisons, like say you have ethanol, or say you accidentally got poison ivy, or say you accidentally ingested some, I don't know why you would, but say some formaldehyde, okay? You have a poison inside your body, you got rid of it. What if you get bitten by a snake or a spider or some kind of insect and it injects some venom in you? You have to get rid of it. What if you have some kind of toxin in your body? Okay. What if you take an illicit drug or even a regular drug and you need to eliminate that drug from your body? Okay, so we use a lot of different types of reactions. We're only going to focus on metabolic in Unit 2, but there's a lot of more interesting reactions. Some other ones include breaking down glucose to form ATP. We're going to talk about that in metabolic, but it's not going to be one reaction. It's going to be a huge series of reactions. We break down ethanol, we can break down a venom or a plant poison, and the activation of certain medications. Some medications come inside our body inactive, and we have to have our body actually turn them on. Okay. So this is what I'd like you to do today, okay, or tomorrow, and we can get it done in the next couple days. I'd like you to research at least 4 reactions that the body carries out that is controlled by an enzyme. Make sure it's one that's controlled by an enzyme. So in your research, I'd like you to report the following. Okay, I want to know what the products are, what the reactants are, and what the enzyme is used. Okay, I'd like you to see a picture representation of the reaction. Okay, and I'll show you just what I mean here, like not this, because these are words, but more of like here. Okay, I want to actually see what the molecules look like. I'd like you to mention where it occurs in the body, like does it occur in the liver and the muscles. It might be tricky. I mean, it's not imperative that you have that, but it's kind of neat to know where that is. A good thing is to see if you can guess how the reactants would get into the cell. If you're going to have this reaction, it's most likely going to happen inside a cell, not always, but most of these reactions are going to happen inside a cell. So that means the reactants are going to have to get in there somehow. How are they going to get in there? Is it going to go through active transfer, passive transfer, facilitated fusion? It's going to use a channel. It's going to use a carrier. Okay? If you can figure out which organelle carries out that reaction, once it's inside the cell, who's doing it? Is it the Golgi apparatus? Is it lysosomes? Is it peroxisomes? Okay. And then I'd like you to, I'm not going to mark this. Okay. What I want you to do, this is kind of like what you would do for homework normally, but I want you to send this to one of your study partners so that you can have kind of 2 lists of reactions and you can compare and say, okay, I completely get this. This was a great reaction. I really like this one. I'm going to be able to remember this reaction for the future. The key is you want to have reactions that you remember or that you enjoy because they're going to make a lot more sense. So if you're really into venom or if you're like, oh, that toxins are really neat, you may want to look that up. So here's an example of the breakdown of alcohol. Okay, so here's 2 reactions here. So here's my first reaction. So when you drink ethanol, okay, ethanol is going to be converted to acetaldehyde. Okay, it's going to use a series of different enzymes. So if you look this up, you don't need to tell me every single enzyme. Just pick one enzyme that is used to convert alcohol to acetaldehyde. Okay? Now, acetaldehyde is a very poisonous thing in our body, so it's got to have to go through another reaction. So we go through another reaction using an enzyme ALDH, and that's going to convert it into acetic. Sorry, had trouble pronouncing that. Okay, so there's two reactions that you're going to go through. So you could mention here's the reactants, here's the products, here's the catalyst. I like a picture representation. You could say where this is going to happen. It's going to happen in the liver cells. You know what exactly organelle is going to carry it out. Okay. So that's what I'd like you to do today. You're not going to be sending this to me though. Okay. Again, you're going to be sending it to one of your partners. As tomorrow, we're going to move on to a little bit more information on basically how reactants work. Or sorry, how enzymes work, and that will be it. Now again, we're going to have our unit test probably a week from today. Okay, so I just wanted to mention that now so that you know you're ready, because you will have to log on for that week, and I'll be giving you lots of prep work before that. Okay, so